FutureTalk. In recent years, we've seen enormous progress in the biomedical sciences. We've made huge strides in fields like genomics and stem cell research, but we've made a lot less progress in understanding the social implications of these advances. On today's program, we're going to talk about where these advances may be leading us and what kinds of challenges they may be confronting us with. My guest is Hank Greely, professor of law at Stanford University and director of the Stanford Center for Law and the Biosciences, where he specializes in the legal and ethical implications of new biomedical technologies. He's also chairman of California's Human Stem Cell Research Advisory Committee, and he sits on many other boards and commissions that deal with the issues raised by biomedical research. He's an elected fellow of the American Association for the Advancement of Science and a professor of genetics by courtesy at Stanford Medical School. Hank, welcome to the program.
Thank you so much for having me on.
Hank, what do you think is the most important work taking place in biomedical technology today?
Well, right today, I would say it is artificial hips because I got one 8 weeks ago and I love it.
Well, congratulations.
Thank you. But more generally, I think probably the most exciting work right now is a tie between genomics work, understanding DNA better, and the neuroscience work. I'd give a slight edge to genomics because it's closer to fruition than the neuroscience work is, but they're both mind-blowing.
Now, what's happening in genomics right now that we should be concerned about?
So let me start with the two things that we should be impressed with. There are two technical revolutions going on that open up all sorts of possibilities, some for good, some for not so good. One of them is the increase in our ability to sequence DNA. Your DNA is a book made up of 6.4 billion letters. Every letter is an A, C, G, or T. We first sequenced a whole human genome 12 years ago, and it cost $500 million. Now you can go to Mountain View and have that genome sequenced for about $1,500.
And sequencing means that you're reading the genome and you know everything on that genome?
Yep, you're reading all 6.4 billion of those A's, C's, G's, and T's, which, by the way, is like reading the Lord of the Rings trilogy 1,500 times.
Do we know what all those letters stand for? For example, let's say there's a gene that controls your eye color. Do we know where that specific gene is, and is it always on the same place in every person's genome?
Yeah, pretty much. When there are genes whose function we know, they're almost always in the same place, and they're almost always exactly the same. Any two humans on this earth are 99.9% genetically identical. So the genetic differences between us are relatively few. They're not in what genes we have, usually. They're in what versions, what variations, what flavors of the genes we have, and that's determined by the spelling of the A's, C's, G's, and T's inside each of those genes.
So if you take one cell from a human being and then extract the DNA and examine it, you could describe all of those persons' physical traits even though you've never seen that person?
Well, that's sort of the catch. So we've had a great revolution in reading the sequence, but we haven't had such a great revolution in understanding what it means. Some things we understand. So if you've got the wrong version of a gene called Huntingtonin, you will get Huntington's disease. In fact, the only way you will not die from Huntington's disease is to die first from something else. So there's some things that are really heavily determinative. Your blood type, AB, O, or AB, completely determined by your genes. Most of life, though, is more complicated. Things like common diseases, diabetes, schizophrenia, the cancers. For most people, genes play some role, but they don't play that big a role, and we don't yet understand even the genes that control the small role that genes actually play in those common diseases. So it's good news, bad news. A few things we understand really well. Some things we're beginning to understand. Some things we've got no idea.
But do we expect to understand it eventually, given enough time and resources?
So that's the $64 trillion question, or $6.4 billion question for the base pairs. Some of it we clearly will, I think, understand. Some of it we never will, I believe. Things like the details of your intelligence are going to be so affected, not just by your genes, but by your environment and by chance, that we'll probably never be able to say, this embryo, it's going to grow up to get a 2350 on the 3-part SAT.
So there are a lot of intangible qualities that
Right.
you can't trace to genes.
Well, there's genes that influence them. We might be able to say this embryo's got a 60% chance of being in the top half, but a 40% chance of being in the bottom half, and beyond that. So that's at one extreme. Something like Huntington's disease is at the other extreme. How far we'll be able to move between those, how well we'll be able to predict who's going to get type 2 diabetes, who's going to get lung cancer, and so on, is an open question. People are optimistic, but optimism's kind of a requirement for life and science.
Now, reading the genome is one thing. Can we go the next step and modify the genome?
So that's the second revolution that's going on. So the first is the revolution in reading and sequencing the genome. The second is the revolution in editing it. And that one is almost brand new. But that's not quite fair. Back in the early 70s, scientists here in the Bay Area, Paul Berg and Stan Cohen at Stanford, Herb Boyer at UCSF, first learned how to move genes from one place, from one organism to another. And Berg won the Nobel Prize, and Cohen and Boyer got a patent that made them rich. But to move and to edit these things has been expensive and difficult and not very effective. We've had ways of doing it. It's like cars before the Model T. There were cars, they were expensive, they broke down a lot, and most people didn't have them. 3 years ago, scientists, including one at Berkeley named Jennifer Doudna, invented a system called CRISPR-Cas9. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats, and Cas9 stands for CRISPR-associated protein 9. I don't really understand it, but it has made gene editing much, much cheaper, faster, and more successful. It's like the Model T. It's the inflection point that's turned the corner for genome editing, and now pretty much anybody with a few $1,000s, a garage, and a college degree in molecular biology or biochemistry can use the CRISPR system to start editing genes.
Now, what do you mean by editing genes? Does that mean that you change the qualities of a specific gene, or you take away an old gene and replace it with a different gene?
Yes, it means both. So CRISPR, the main thing CRISPR does is take out old stuff. So let's say that you have the Huntington gene, the Huntington version that causes Huntington's disease.
It causes it, or it fails to create a defense for it?
Well, this one actually causes it. And so in order to fix that, we'd have to go in and take the sequence that causes, the bit of sequence that causes the disease out of your cells. CRISPR can do that. It can take that out. But you can also combine CRISPR with something that then adds something in. So we could take your Huntington sequence out and we could put a normal sequence in there for you. It's a little tricky for you because as a grown-up, you've got about 100 trillion cells, and making CRISPR work in each of those 100 trillion cells is going to be tricky. If you try it with a baby, it's a little easier. If you try it with an 8-cell embryo, it's a lot easier because there are only 8 cells you've got to change. If you try it with an egg and a sperm, easier still. Only 2 or maybe only 1 cell you've got to change. So CRISPR is a real turning point in making genome editing as cheap and easy as genome sequencing is beginning.
Now, can this be done inside the body, or is this all done in a bot? Like, let's say you want to perform some procedure on an embryo. Do you have to have the embryo out of the body in the lab to do that?
So you can do this in bodies. It's not as easy. For an embryo, you'd probably want it in a lab, which is why this would probably be done in conjunction with in vitro fertilization, where the embryos are in labs and not in a fallopian tube someplace. But for some purposes, particularly for dealing with non-humans or for dealing with certain human diseases like sickle cell anemia, which is a disease caused by genes as they work in the bone marrow, or as actually they don't work right in the bone marrow. You could figure out ways to change the bone marrow in the person's body, or you could take some bone marrow out, change that, and put it back in. So you could do some of it in vivo, in bodies, or some of it in vitro, in test tubes.
Do you have to do one cell at a time, or is there a procedure where you can do many cells at once?
You can do many cells at once. We still can't guarantee that all of the cells will take, and we still can't guarantee that it will always go in the right place. But the advantage of CRISPR-Cas9 is it's better at both of those things, much better at both of those things than what we used to do.
So you can remove a gene or a sequence of genes, replace them with new genes, and they'll attach seamlessly so it'll be as if they were always there?
Yep. Well, except to the extent they've already done damage to your body, that damage doesn't necessarily magically go away. But moving forward, it's as if they've always been there.
So what kind of things, once this technique is really perfected, what kind of things could we change? Presumably, if there are genes that make you more prone to disease, maybe we could fix those. But could we have overall improvements in a person, make people taller, stronger, smarter, all the desirable qualities that people might want?
So yes, no, and maybe. We should be able to change some of the traits, but some are going to be easier to change than others based on how much we know about them. As I said earlier, some things we know a lot about, some things we know a little about. I think it's useful to break them down into 4 categories. Diseases, we should be able to change the ones we understand, like Huntington's disease or sickle cell anemia, or some breast cancers, those that come from defects in BRCA1 or BRCA2, 2 kind of famous genes. The ones we don't understand, we can't help. Cosmetics. We should be able to change a baby's eye color eventually, or whether a man will have either male pattern baldness or very young white hair. Behavior. I don't think we're going to be very good at this, but we should be able to say 60% chance of being in the top half in sports or music or math. And finally, the easiest one, although trickier to change, but if you start early enough, you could do this too, boy or girl.
Now, where do you get the new genes from? Let's say you're replacing some old genes with new genes. Do you have to get the new genes from a tissue sample of another person, or are they simple enough chemically that maybe you can synthesize genes in the lab?
Yes, so for over 15 years, you've been able to go to a website, several websites, put in the sequence you want, give them your credit card number, and a few days later, a test tube shows up with that gene sequence in it. And you just use their companies who make their living synthesizing stretches of DNA for you.
Now, some people are a little bit troubled by the idea of changing human genomes. What are some of the ethical issues of this? Is there anything that can go wrong? I mean, you're just trying to make people better. What could be wrong with that?
Well, any place there are humans, there's human error. So things can certainly go wrong. We've actually tried a version of this that we call gene therapy for about 35 years. Hasn't really worked yet. There's no gene therapies approved in the United States by the FDA yet, although it's getting close. But in a very famous and sad case, an 18-year-old volunteer died as a result of a gene therapy experiment in 1999, Jesse Gelsinger. For him, it went wrong. So things can always go wrong. I think what people most worry about fall into a couple of categories. One is the safety. And again, you know, if you're making a baby this way, so if we did gene therapy on you or me, we're consenting, presumably competent adults. We decide, I'm willing to take this risk because I really want to avoid this disease or cure this disease. If it goes terribly wrong, some of our friends would be sad. But in the long run, we made a decision. It didn't work out. We're going to die anyway. It's not such a tragedy. If you make a baby using this and something goes wrong, the baby never volunteered for it. And that baby might have 60, 70, 80 years of terrible life because of your mistake. So making babies this way, the safety risks are serious. People, I think, appropriately worry about some of the fairness risks. If this is expensive and only rich people can get it, that may be kind of an ugly world. If this leads people to choose to make or have boys rather than girls, and we get a sex imbalance as exists in a number of countries around the world using more old-fashioned methods like ultrasound and abortion, that could be bad. And I think in some ways the toughest one of these equality issues involves people who currently have a genetic disability. If you have a child, let's say, with Down syndrome, and these techniques make it likely that no children will ever be born again, or very few children will be born with Down syndrome, how does that affect your child? His chances for research to work on his disease, his social support, the sort of stigmatization he gets. I once had a young woman who had a disease called spinal muscular atrophy that put her in a wheelchair. And it's a genetic disease. It gets worse as she ages. She was a Stanford grad, brilliant young woman. And she said, you know, this system you're talking about means I should never have been born. And I wanted to say, no, it doesn't really mean that. But in a way, it does say that her parents could have had a choice of having her with the disease or having another baby without the disease. So I find that really troubling.
Yeah. I mean, should we hold back progress ways of benefiting other people because there are some people who wouldn't be eligible for those benefits. Would that be fair to the people who might still receive the benefits?
It depends on what you think of as fair.
Yeah. There's also,
and that's an argument philosophers and theologians have been having for a long time.
There's also a legal issue. Let's say that they perform this procedure on a baby and the wrong gene goes in the wrong place and he's horribly deformed. Who's going to pay his medical bills for the rest of his life?
The doctor's malpractice insurance, most likely.
Which will probably put a lot of doctors out of business.
You know, that's an issue that the medical profession's already confronted. OBGYNs have some of the highest malpractice issues, malpractice premiums in the country. The other sort of issue people are worried about is less tangible. You've got safety, you've got fairness, there are issues about coercion. What if the government says you can or can't do this or that? The one I think bothers most people is, to put a not very precise term on it, playing God. Are we playing God? Are we wise enough to make these decisions about the genes for our descendants? I've never been particularly taken with that argument. I think we clearly aren't wise enough to make perfect decisions, but that's not really the question unless you really believe that there is a God who's making all those decisions for each child. From my perspective, it is, are we better than chance? Are we better than the random mixing of genes that fertilization the old-fashioned way, making babies the old-fashioned way gives us? And I think we can hope to be better than chance, although we can't be sure of it.
Are we doing this for the sake of making human generations better, or maybe people are doing it for their egos to see, look, the great things that I can achieve? Maybe that's not the best motive to do this kind of research.
So I think there are, well, research motives are complicated. People are doing it to try to relieve human suffering and to try to get tenure and to try to win the Nobel Prize. Motives are hard things. But I do think, for whatever motive, in the next 30 to 50 years, we're likely to see new ways of making babies. One of them will be genome editing. One of them, which we haven't really talked about, but which I think is going to be most important, is genome selection. You make 100 embryos. Because sequencing is so cheap, you test the genomes of all of them, and you say to the parents, which one do you want to move? And then a third way, and this plays into your egotism, but at a different, your egotism point, but at a different level, I think human reproductive cloning is probably going to happen sometime in the next few decades. So talk about egotism, you want to make a baby that's just like you. Human cloning, or just like somebody else you care about, human cloning gives you that opportunity. I used to think that wasn't going to happen. In the 15 years, 16 years after Dolly, nobody had succeeded in cloning a human embryo, despite lots of efforts. 2013, a group at Oregon did. So I think cloning is back on the table.
Now, to clone an embryo, let's say an embryo just has one cell initially, and it splits. Could you separate the two pieces, and now you have two identicals?
Yeah, people have done that, not in humans, but it could be done in humans. That's the way identical twins happen. At some stage, naturally, before the embryo gets too deeply implanted in the uterus and the pregnancy gets too far gone. So in the first 8, 9 days after fertilization, it splits in two. They've got the same genes. They're identical twins. But of course, if you've known identical twins, you know that even though they got the exact same genes, they're not the exact same person.
Because maybe they want to be different. Maybe they both have a desire to not be like the other one.
You know, maybe, but I think it actually goes back. They're more like each other than any other two people will be like each other, but they're not the same. And I think that speaks to the importance of environment, of chance, of all sorts of factors we don't understand that affect, in some ways, not in every way, but affect how the genes play out. They won't affect what your blood type is, but they could well affect your intelligence or your susceptibility to a bunch of diseases.
Now, there are some people who call themselves transhumanists who say that we can modify our own evolution to create humans that are more than human, that have attributes that no other humans have today.
Yeah, they've actually changed their name. I don't know whether this will be a good marketing strategy. They're now Humanity Plus.
Humanity 2.0?
Yeah, so my clear and strong answer to that is maybe and maybe not. We don't understand very much about what makes, genetically, what makes people better. We know a fair amount about genetic variations that give people very low intelligence, pathologically low intelligence. We know basically nothing about genetic variations that give people higher intelligence. So first we got to figure out what the enhancing variations are, if there are any, and then convince people to be willing to use them. We may be able to change some things like eye color and hair color, but intelligence, math ability, musical ability, again, I suspect the best we'll be able to do is a 60% chance of being in the top half.
And we're still talking about changing embryos. We're not talking about changing adults.
No, I mean, although, you know, we do things that enhance born people. The greatest cognitive enhancing tool that humanity's ever come up with is reading and writing. And not only do we allow people to teach their kids reading and writing, we require it. That's a way of changing somebody's cognitive abilities after they're born. You don't change their genes at all, but you change them fundamentally. And I think that leads to a really important point. People get very excited about genetic enhancement and genes enhancing. You know, genes are significant. They're not magic. The difference between the best genome we could probably come up with today and average, I'm willing to bet would be less than the difference between a kid born to a wealthy family in a rich country and a kid born to a poor family in a poor country. And those are completely environmental differences that we could fix now, but we don't.
I understand there's a lot of work being done in stem cell research these days. First, could you just give a very brief description about what is a stem cell compared to other cells?
Sure. There are two interesting things about stem cells. One is they will live forever, or at least for an indefinite period. Most of your cells will divide 40, 50, 60, 70 times and then die.
I mean, the stem cell lives as long as the individual lives?
Yes. So we know that they can live at least 123 years because the oldest known human lived to be 122 and a half. And so you add on the 9 months for pregnancy, 123 years. So they don't die off after a limited number of cell divisions. But the other thing is they will turn into different cell types. So an embryonic stem cell, you and I started out as a small number of cells in the middle of a 5-day-old embryo, poetically called the inner cell mass. Those cells which make up human embryonic stem cells can make every cell type in the body. We know that because they did, for you, for me, for all of your viewers. What makes it exciting is you could then take those cells and make them into heart muscle cells, brain cells, kidney cells, in a way that adult bodies don't do. So that's the important thing about stem cells.
How do you tell it to do that? Every stem cell has the capability to become any specialized cell, but how do you make it become the one you want?
As always, it's a little more complicated. There are stem cells that are halfway down, so there's blood-forming stem cells. They make all 100 different types of blood cells, but they won't make kidney cells or brain cells. So if you start with a blood-forming stem cell, you're going to get a blood type, a type of blood cell. Which type do you get? That's what the research for the last 15 years has been focusing on, with some success, but not perfect. Basically, you put them in environments where they're getting chemical signals from other cells that tell them this is what you should become.
Now, in a baby that's gestating in the womb, for example, every cell seems to know its place. You have the right amount of muscle cells and the right amount of bone cells, and they're all in the right place.
Well, if you get far enough to actually be born alive, most pregnancies, most fertilized cells never become babies. Most fertilized eggs never become babies. Maybe because something goes wrong, and maybe it's something that goes wrong with that system. So early on, an embryo divides into, the stem cells divide into 3 different cell types that make different kinds of tissue. And then later on, if you're in the part that makes a brain cell, where it is in the brain and what signals it's getting from the things around it help determine what kind of brain cell it turns out to be. But we're still trying to figure that out. That's where the frontier of stem cell research now is trying to make these cells become what we want them to be. We can do it for some. We can do it occasionally for others. We're having trouble doing it at all for other types.
Let me ask you this question, which is a slightly different topic. So I've heard that it might be possible to revive long extinct species. Like maybe you could find a dinosaur bone and find a few strands of DNA in that and somehow manage to merge that into a live cell of some creature and grow new dinosaurs.
Maybe you could revive liberal Republicans.
Well, I wouldn't go that far.
Dinosaurs are out. I don't know about liberal Republicans.
Okay.
We're not going to be able to do dinosaurs. The last dinosaur, unless you count birds, which are kind of surviving dinosaurs, died off 66.4 million years ago. No DNA is going to be left. The Jurassic Park trick of the amber was clever. People have looked, doesn't work. But Pleistocene Park, that's really quite possible. We have done whole genome sequences of animals that died 200, 300,000 years ago during the Ice Age. The DNA is preserved, so we know their genomes. We know the genome of the woolly mammoth. And people are talking about reviving, maybe not exactly mammoths, but something like mammoths by taking cells from an Asian elephant. Turns out mammoths were closer to Asian elephants than African elephants. And using CRISPR-Cas9, which we talked about earlier, editing those cells, sequence the Asian elephant, sequence the mammoth, see where they're different, edit the Asian elephant cells to make them like mammoth cells, and then turn that into a baby and hope that an Asian elephant mother will give birth in a very confused way probably to a really hairy little elephant calf that's a mammoth.
Do you think like this process would need to be regulated somehow, or people who could afford it should just go ahead and
No, I think there should be some regulation. I don't think we should be terrified of it, but you wouldn't want to be the person who unleashed a new invasive species on the planet. Kudzu is a real problem in the southeast. Clams, the Chinese clam is mussel is a problem in various places. You don't want to do that, so you need some regulation before there's environmental release. And you also don't want to kill off too many mother elephants. You want to make sure this is safe for some of the animals involved.
We just have a few seconds left, so let me ask you this last question. Are you optimistic that all this research is going to lead to something good for humanity in general, or do you think maybe we're treading on dangerous ground? And you have about 5 seconds to answer.
I'm optimistic. We will screw up occasionally, but on balance, this will be a good thing.
Okay, very good. And on that note, I'd like to thank you for being here today.
My pleasure.
Hank Greely, Director of Law and the Biosciences at Stanford University. I'm Marty Wasserman for FutureTalk. Visit our website, www.futuretalk.net. For FutureTalk, we'll see you next time.